applied to the hippocampus could induce long-term potentiation and depression of the EPSP. When you apply some of these protocols at lower frequency than they did here, but when you apply repetitive TMS to the motor cortex, it's a little bit trickier. It's a little bit less clear. Some studies show that 5 Hz rTMS applied to the motor cortex can increase the motor evoked potential on the left side and change EEG patterns on the right side. Other studies are less clear. Other studies show that 10 Hz rTMS on 2 different days really don't affect motor evoked potential that much, whereas 1 Hz does depress and 20 Hz does increase the motor evoked potential. And more recent studies looking at continuous and intermittent theta burst stimulation, which is thought to be inhibitory and excitatory, respectively, across a large number of subjects, really don't have a net effect. So even in this space in humans where we have the motor evoked potential, which has very well developed electrophysiology and high signal to noise, there is not that much consistency on a subject to subject basis of these different patterns. And outside the motor cortex, we have even less evidence of what happens. What do we think TMS does outside of the motor cortex? Well, we think one hypothesis is that in depression, you have a hypoactive DLPFC, and that translates to a hypoconnected frontoparietal network in blue. That this decreased executive function has a weakened top-down inhibition and allows the default mode network to be hyperconnected and run awry, and this can underlie ruminations and other symptoms in depression. When you apply repetitive TMS at high frequencies, or at least at 10 Hz, you can activate the DLPFC or induce long-term potentiation of the DLPFC, which can strengthen and normalize the frontoparietal network, normalize the top-down inhibition of the default mode network and other networks. This is one hypothesis. There's some evidence in the imaging literature on this, but I would not say that there's reproducible evidence to suggest that this occurs consistently and that this relates to clinical symptoms. So my goal is that when I approach a patient, any patient, and any brain circuit, that we can take TMS and break it down into its most fundamental pieces so we can develop a set of stimulation parameters that will strengthen and weaken that specific circuit for that individual at that time period. And if we can do that, we have a treatment with really unlimited applications and minimal side effects. TMS, as people know, is approved for depression, but is also approved for many other neuropsychiatric disorders. And my prediction is 5 years from now, it will be cleared for every neuropsychiatric disorder. The worry is that this is all open label treatment, mostly using 10 Hz or theta burst applied to the DLPFC. And my worry is that we're underutilizing this treatment and that we can boost the effectiveness of these treatments considerably if we take into consideration one's physiology, connectivity, and develop adaptive treatment to better change the brain and alleviate symptoms. So what's stopping us right now? I think there's a couple things that are real limitations to moving to this personalized approach. The first is the development of an objective biomarker. We really don't have a single measure or multiple measures that measure circuit excitability that relate to clinical symptoms. If we had this, we could monitor this in real time, monitor these features in real time. We'd have a good idea of how treatment is changing the brain, how treatment over weeks would change clinical symptoms, and taking this target engagement approach would allow us to then adapt treatment if we needed to early on. We lack the understanding of this multidimensional treatment space, right? So we talked about 10 Hz, 1 Hz, and theta burst stimulation, which we know a little bit about, but there's a ton of unexplored space here. And in this multidimensional parameter space, you have location, frequency, pattern, amplitude, duration, state dependence, number of sessions, intersession spacing. I really could go on here. The idea is to be able to move a little bit more towards this multidimensional tuning space where you can identify how certain parameters interact and relate and maximize a biomarker. Resources are a huge issue here. When you do clinical trials, if we're comparing 10 hertz to 9 hertz treatment, right? We'd randomize 40 patients in 10 hertz and 40 patients in 9 hertz. We'd have almost 100 patients. We'd apply a month long of treatment. And that adds up to almost 3,000 TMS sessions. It's really, really difficult for a small lab and even a large lab to handle many of those clinical trials. So we need to take this number of subjects and amount of time, space, and shrink it down so we can do these studies quickly and efficiently. We think we can get this 30 days of treatment down to a single day, just in terms of studying how different stimulation parameters affect brain circuitry. And these n's of 100, let's get it down to an n of 1. Let's do crossover design studies, and let's explore quickly this enormous parameter space. And finally, once we have the above, we can start looking at developing real-time solutions and adaptive solutions to update the treatment in real time. And I'll dive into this in a bit. So this is our lab, started about a year ago. We focus on neuromodulation and human electrophysiology. Our mission is to deconstruct brain stimulation to establish the fundamental mechanisms underlying human neuroplasticity and develop real-time closed-loop treatments for mental health disorders. We do this 3 different ways. One is to develop novel methods to probe the human brain in a causal and directional manner. The second is to better understand the mechanisms underlying human brain plasticity. And the third is to develop these real-time closed-loop solutions. We use 2 different preparations to study this in humans. The first is an intracranial EEG approach, and these are in patients with medically intractable epilepsy. I'm not going to dive into this approach much in this talk, but we've done a considerable amount of work here. I'm happy to talk to anyone offline about this. But basically, we have the ability in these epilepsy patients to stimulate very specific parts of the brain. We can use electrical stimulation and stimulate electrodes that are located in the DLPFC, let's say. And then there are electrodes in hundreds of different brain regions, and we can measure the brain's response to these novel stimulation patterns that we may test out. So we have really nice focality and really nice signal to noise, as well as high spatial and temporal resolution. The issue with this approach, of course, is that these are in epilepsy patients and that this is electrical stimulation. So the talk will be focused on the TMS EEG approach. Fully non-invasive, can be applied in the clinic with healthy controls or depressed patients, where the issues here are focality of stimulation and spatial resolution of the EEG brain response. So we'll dive into this a little bit more. Another limitation is the scalp and the signal to noise in the EEG recordings we get. So the hope is that we can bounce back and forth between these 2 models, is that we can explore novel treatment patterns using the intracranial approach, and then we can see if we identify and can observe these signals that may relate to plasticity non-invasively in a generalizable way using TMS EEG. If we see a signal in TMS EEG, we can look at the neural correlates intracranially. And actually recently we have a paper that's hopefully getting published in a week or so in bioRxiv, not published, but uploaded to bioRxiv, combining these approaches. So comparing adding TMS intervention in these epilepsy patients where we have intracranial EEG recordings. Okay, so how do we get to this next generation of brain stimulation, which will be personalized, which is not that new. Right now we personalize based on dose in the clinic today. There's lots of personalization research on targeting, but then also adding adaptation. Can we update the stimulation in real time? And that is pretty novel in terms of treatment for depression. So our current open loop treatment is 10 hertz with theta burst stimulation, no recording of EEG, really blind to one's physiology and plasticity. Our newer adaptive approach will be starting with a fixed stimulation pattern, but because we're measuring the EEG brain response, we can see how plasticity is induced, how strong is that change, how specific is that change, and we can update the stimulation pattern accordingly. So this we'll focus on the TMS evoked potential, which I'll dive into, and specifically the P30, the first evoked potential that we see. But we can adapt treatment based on this biomarker. It's based on one's physiology, and hopefully it's faster and much more effective. So if we zoom out and look at the 30,000 foot view, treatment, when I go into the VA and treat my veterans, usually targeting is not based on an MRI, but it's based on a BMF3 approach of an estimation of where the DLPFC is located. Dosage is through the motor threshold, multiplied by 1.2 to account for the increased distance in the DLPFC between the scalp and the cortex. And treatment is open loop on a daily basis, 30 treatments or so. Research in the field, there's targeting is a hot topic. John Siddiqui, Ellie Cole, Nolan Williams, Robin Cash, and others have been focused on a resting fMRI approach to identify the negative correlation, and Alison Rosen as well, to identify negative correlation between the DLPFC and the subgenual cortex. The idea is that if you can find that location and boost that part of the DLPFC, you will have some nice top-down inhibition of the subgenual cortex and normalize that subgenual hyperactivity that seems to exist in depression. Dosing, interesting research around dosing involves accounting for the individual's distance between skull and cortex and adjusting the treatment dosage as such, and also electrical field modeling to see how much of the TMS pulse gets into that part of the DLPFC for that individual. And treatment, the big research around the treatment is adding more treatments in a given day. At first, this was twice daily treatment, and then 3 times, and recently Nolan Williams and colleagues have applied 10 treatments a day with really nice clinical results. So can we get this whole treatment down to a week? Our approach is slightly different. We do focus on targeting, but we really believe in this idea of having an inexpensive and clinic-ready approach. So we think we can measure these brain circuits using TMS EEG in the clinic without the need for fMRI. Dosage is typically thought of around power, but like I said before, we have multiple different parameters here. So can we take this multidimensional space and identify that peak that modulates the biomarker that we think relates to depression? And then use that for treatment. Use that as a treatment start, but then also update the stimulation in real time. And we can do this every day. So the 3 things that we need to get there is development of a brain biomarker to track this brain change, which relates to symptoms. Understand how these stimulation parameters affect brain dynamics, and develop real-time monitoring and closed-loop stimulation approaches. We're going to focus on TMS EEG in this talk, which is not developed by us, but it is a really nice non-invasive way to get causal perturbation of a brain region, millisecond temporal resolution of the voltage response at many different brain regions, and is clinic ready and relatively inexpensive. It's got tons of artifacts. It's got pulse artifact, decay artifact, various other EEG artifacts that need removal. And so our group, as well as other groups, have developed different ways to clean that TMS EEG signal. In the end, I'm not going to dive into it, but in the end, you end up with this TMS evoked potential. Time 0 is when a single pulse of TMS is applied, and each line in blue is a channel. So you're seeing the 64 channel response to a single pulse of TMS, repeated 100 times in average, because a normal single pulse EEG response is pretty low signal to noise. So you see this multiphasic response that lasts about 300 milliseconds. It has multiple different peaks. Each of these peaks in the arrows have interesting spatial topography. As you can see up above, 30 is 30 milliseconds, 40 is 45 milliseconds. The bottom line is if you plot this on a brain and make a video out of it, you get these nice videos of causal and directional brain dynamics, all non-invasively. So the question is, can we use this TMS EEG approach to find a biomarker with high signal to noise that measures network excitability, is quantifiable on a millisecond basis, and changes with TMS treatment. We think we've identified one of these biomarkers, and we call this the P30, as has been called in the field. Using TMS EEG, single pulse of TMS provides this waveform. This first positive potential is the P30. P for positive, 30 is around 30 milliseconds, usually 25 to 35 milliseconds. It is relatively lateralized to the left side. We know that this P30 is thought to reflect local excitability based on some pharmacology studies that showed that when you add in voltage-sensitive sodium channel blockers, it suppresses the P30. And this is really the first potential that you can see cleanly outside of motor cortex after the TMS pulse. We also see this potential intracranially. So this is our electrical stimulation approach in epilepsy patients, where we apply the electrical stimulation pulse to the DLPFC. So the patient doesn't know and doesn't feel the stimulation, which is important to suggest that this is not sensory. And we see a 20 to 40 millisecond response. Now there's 3 lines of evidence to suggest that this P30 may be useful as a biomarker. The first is a randomized clinical trial that was run in Amit Etkin's lab, where we randomized patients to either active TMS, active 10 Hz TMS, or sham TMS for 4 weeks. And we measured before and after those 20 days, single pulse TMS EEG. We looked at each of the potentials in the TMS evoked potential. We looked at P30, N45, P60, and really only saw statistical differences in the P30 potential. So what we saw was a group by time interaction, group being real versus sham TMS, and time pre versus post treatment. We saw that this effect was localized right around the stimulation site, so left prefrontal region as well as left parietal region. When you break this down, we saw a main effect of time in the active but not the sham treatment group. When you look at the P30 and ask what happens, the P30 is actually getting suppressed after active but not sham stimulation. This P30 is localized, seems to localize to the medial and lateral prefrontal cortices. And although the correlation is weak, it seems like there might be a relationship between the amount of P30 suppression and the amount of clinical improvement, albeit the very low sample size here. The second line of evidence is in a healthy control study where we applied 10 Hz TMS. We had 1 day of active TMS and 1 day of sham TMS. And we saw in the active but not the sham day, active repetitive TMS suppressed this early evoked potential here in the parietal region, and we didn't see any changes in a nearby visual region. The last piece of evidence here is intracranially, where we applied a single day of 10 Hz electrical stimulation to the DLPFC. Before and after that 10 Hz stimulation, we applied single pulses. So here's a single electrical pulse to the DLPFC, measurement in this parietal site at the circle. You can see these nice single trial evoked responses. And a single trial is actually 20 to 40 milliseconds. When you look before versus after the 10 Hz electrical stimulation, you see a suppression of this 20 to 40 millisecond or 30 millisecond response that lasts for about 20 minutes after the stimulation. This effect is localized in the left prefrontal and parietal region. So we think we have a signal that can be identified both in intracranial EEG and scalp EEG, gets suppressed after a single day and 4 weeks of 10 Hz stimulation, and may relate to clinical outcome. Now the big issue with this P30 is signal to noise. Although it's the first potential right next to the TMS pulse, which from a mechanistic point of view is really exciting and can be interpretable, it's got low amplitude. So usually TMS EEG studies show between 0.5 and maybe 2 microvolts around the P30. and this is averaged across 80 or 100 trials. So when you think about using this biomarker in real time on a single trial basis, it's almost impossible the way it's evaluated. So our next set of studies are going to be focused to boost the signal to noise of this P30. To boost the signal to noise of anything, you can either reduce the noise or boost the signal. So we're going to start by trying to reduce the noise. So this is a study spearheaded by Jess Ross, a postdoc in our lab, and Manjima Sarkar, a research assistant. And the idea is that TMS induces off-target sensory effects unrelated to the direct effect of TMS. Most people think of this as the N100 and P200 time periods, but actually the auditory evoked potential can really start as early as 30 milliseconds. Jess sent me this paper. This is a piano tone, and the EEG responds to this piano tone. And you can see some responses much earlier than 100 milliseconds. So in order to get sort of sound interpretation of this P30, we have to remove these off-target sensory effects. So I was the first subject in this study. This was 21 healthy controls. The goal here is to minimize the central N100 and P200. Now, standard masking procedures to reduce this off-target sensory effect include foam, which can increase spacing between the skull and the TMS coil and reduce bone conduction, and then noise masking. And noise masking is usually in the form of using the TMS click sound, making noise out of it. So always playing in headphones that TMS click sound so that when the actual click comes in, the saliency of that click goes down considerably. We're exploring two other considerations here. The first is the addition of earmuffs or over-the-ear protection, or what construction workers will use. It's passive noise reduction. The idea is that if we can reduce the auditory click that comes in before we have to mask it, it'll be easier to mask. And as everyone knows, it's pretty hard to fully mask the auditory sound, even with a good noise masking. At 100%, it's very intolerable, and it doesn't fully mask that sound. So if we can reduce that 80% and then mask it, it becomes a lot easier. John Rothwell has explored this in motor cortex in his recent paper. There's one sentence in the discussion that talks about the use of it, but didn't explore it quantitatively. And I don't think anyone has ever really used it for the DLPFC. The last piece that we're playing around with is interstimulus interval timing. Timing is interesting. Most people use a jittered interstimulus interval. And the idea there is that you don't want to induce any lasting plasticity effects with your single pulses. But by using a jittered approach, you actually increase the saliency of any individual TMS pulse coming in. Increasing the saliency can affect the N100 and P200. So we actually went back to a more predictable interstimulus interval timing, which reduced the N100 and P200. We also showed that by doing so for 80 pulses of TMS, you don't induce any plasticity effects. So we call this combination attenuate, which is an acronym. Doesn't work that well, but attenuate's kind of fun. And we were able to show across these 21 subjects a really nice reduction in this N100 and P200 using this attenuate approach compared to the standard masking approach, with keeping the earlier potentials, which we're interested in looking at and we think are direct effects of TMS, intact. We were also able to reduce the loudness perception and the scalp sensation using our attenuate approach. So in summary here, we were able to reduce the N100 and P200 as well as perception with no change in the early evoked potentials. So we think we've reduced some of the noise here. Now let's work on boosting the signal. This can be done in two different ways. We took an analytic approach first. And so this was spearheaded by Chris Manassi, a grad student in the lab, Austin Talbot, a postdoc, and Chris Klein, a staff scientist in the lab. The idea here, if you just look at single pulses of TMS to the DLPFC and look at the evoked potentials, each trace here is a different brain region. Not really important, but what I want you to take from here is that in this time period of this P30, it's pretty complicated. And there are standard ways to quantify a peak, but it may not be ideal, and it's only one way to quantify it. And to my knowledge, an exploration of how to quantify a peak in the TEP hasn't been explored in a really deep manner. And there may be other features than this one way to quantify the peak that may be informative. It may be a combination of features or a multivariate approach that in combination may be super informative in, let's say, looking at the separation of an active TMS arm versus sham TMS arm. So our goal is to develop this multivariate approach using machine learning that will hopefully boost the effect size between real and sham stimulation and the signal to noise of the P30 itself. And what we're going to be doing is exploring these following analytic considerations. So looking at different time windows, different ways of peak picking, looking in the time and frequency space and saying which one is better, and region of interest. As you can see, this is a huge space, and even looking at region of interest, there's many different questions that come up. Should we be doing this in sensor or source space? If we're looking at sensor, should we pick 10 channels around the left prefrontal region or 6 channels or 2 channels? Should we pick those channels based on the literature? Do we pick it based on a single subject data-driven approach or a group level analysis? So we're going to explore all of these and throw them in and pick out those features that best separate real versus sham stimulation. Another way to do this, to boost the signal of the P30, is to do it experimentally. So the idea is that if we can get good data with high signal to noise coming in, we'll be better off. And so this is Juha's work just starting up now, who's a visiting scholar who just landed in the US. The idea is to do a grid and angle search around the DLPFC. And I understand the effect of location and angle on the P30. And it's different than motor cortex, where we have a set angle that we have to abide by and set location. The DLPFC, it's not super clear exactly where to hit, and it's not super clear that you need to use that 45-degree angle. So if we can find a better angle and location within a small region that gives us a much better signal to noise of our output, we may be better off. More on this in a few months, hopefully. So we think we have this P30 that's present in intracranial and scalp EEG, reflects cortical excitability, gets suppressed after a single day and 4 weeks of stimulation, may relate to clinical outcome, and we think we can boost the signal to noise by suppressing the sensory potentials and optimizing experimental and analytic approaches. So moving on, this large stimulation parameter space that we need to start exploring. So this is a project that's spearheaded by Chris Klein, a staff scientist, and Jess Ross, a postdoc in the lab. The idea here is if you just look at 10 Hertz treatment, and this is a schematic, each line here is a stimulation train. Each row is a different day of treatment. So day 1, you have 75 different trains that you're applying, 3,000 pulses, and after 30 days, you've applied a whole bunch of stimulation. It's absolutely impossible to compare this approach in an individual to 5 other treatment approaches. It just doesn't work. You can't do it. It will take months. And so we really need to reduce these dimensions. We're starting way too complex. So in more simple terms, which actually haven't really been explored much using TMS EEG, we're asking these questions. Do single TMS trains elicit acute brain changes that we can quantify with EEG? How long lasting are these brain changes? Is it 5 milliseconds or 500 milliseconds or 10 seconds? How do these brain changes differ based on frequency, amplitude, and duration? And then once we've explored a single train, then we can start building up these trains and look at interesting plasticity and metaplasticity effects. So when you add these stimulation trains consecutively, how do these changes build up? Are they linear or superlinear? Do they decrease after you add 5 of them, or do they, is it an exponential increase in the biomarker that we're looking at. So we'll explore 1 Hertz in theta burst, but we'll also explore this very unexplored space in TMS. We talked about these different dimensions. We're going to try to tackle the first 3 first. You can see these are probably ones that make the most sense. And we're just going to look at a single train and ask, using electrophysiology, what does that evoked potential look like, and how does that change as a function of these 3 different dimensions? So we're going to vary the power, duration, and frequency of a stimulation train, and we're going to develop our multidimensional tuning curve for every subject and ask, how consistent is this multidimensional tuning curve on a day-to-day basis for an individual subject? How consistent is it across subjects? What are the different interactions between power, duration, and frequency? Once we have that brain biomarker, once we have that better understanding of this parameter space, we can start to look at real-time monitoring and closed-loop stimulation. So this is a huge group approach. Chris Klein has been driving much of the work in close collaboration with Scott Linderman, who's a statistics assistant professor at Stanford. Chris has developed the methodology to clean and monitor the P30 in real time. We are starting to play around with using closed loop stimulation to quickly identify that multidimensional tuning curve. So can we use closed loop stimulation to map out the important data points within that tuning curve to allow us to quickly evaluate it? If we did just a open loop grid search, it may take 5 hours, but will it take 20 minutes if we can use closed loop stimulation? This is not to induce plasticity. This is just to look at this tuning curve. We're doing this in silico and in vivo to demonstrate proof of concept. Once we have that, then we'll start looking at RTMS and the application of closed loop RTMS. So we'll look at a single session, 10 minutes of stimulation. Does closed loop stimulation perform better than open loop stimulation? Once we have that piece, then we can start thinking about applying a treatment for 30 days or 30 sessions of this closed loop stimulation compared to an open loop 10 Hertz treatment. And this is just one of the first experiments here, is we'll have our open loop standard 10 Hertz RTMS for a single day and evaluate brain changes with our P30. We'll have our tuning curve for that individual for that time period, and we'll use that peak tuning curve to create our open loop optimal RTMS treatment. So this is not closed loop, this is just open loop, but we're personalizing it. And then starting with that optimal stimulation pattern, but then updating that in real time, then we get our closed loop RTMS. The hypothesis here is that closed loop is more effective than open loop optimal, which is more effective than open loop standard RTMS. So I hope from this talk you took from it that interventional psychiatry is super exciting, but there's lots to be done. We need to develop and flesh out these brain biomarkers considerably. We need to understand this multidimensional stimulation parameter space, and we need to start optimizing these real-time monitoring and closed-loop stimulation algorithms. Once we do that, we have this personalized treatment that can really be applied to any brain circuit and any neuropsychiatric disorder. I want to thank everyone involved. This is a huge group approach. Everyone in the lab, the participants, collaborators, funding agencies. Without any of you guys, this would not have happened. So thank you. And with that, we can go to questions.
Awesome. Really, really cool, Corey. Thank you. And I wonder if you can say more about what you think the P30 is. Well, you mentioned networks, so what is your thinking on what network it's an index of? Is this an attentional sort of issue in depression? And why do you need to measure it at the parietal site instead of where you stimulate? And is this an abnormality, or is it a window into something like deeper in the brain that's not necessarily related to P30? Can you just tell us more what you think?
Yeah, and before I do that, should I stop sharing or should I keep this on?
say probably okay to stop.
Let me, I think we're good. P30, yeah. So I think of the P30 as a simple measure of connectivity or excitability. It is, I'd like to look at the 10 millisecond response. I'd like to look at the 15 millisecond response. Right now, with our current recording capabilities, it's tricky. So 30 milliseconds is the earliest brain response that we can discern after a TMS pulse. So I think of it as a general measure of excitability. Where we measure it, I don't think it's specific to the frontoparietal network. I don't think it's specific to the DLPFC. I use it as a measure of excitability. So in a different study, we're proposing to use the P30 in the ACC. Look at medial structures after single pulses of DLPFC TMS. It's really the most interpretable TMS EEG peak that reflects probably not direct, probably some subcortical or cortical projections and oligosynaptic. But this is, action potentials propagating from the local site to some downstream site. So it's capturing the excitability of that brain network or brain connection. You can do that at the DLPFC and look at local excitability, or you can look at other regions. So I don't think it's necessarily special in any way to depression or to the frontoparietal network. I just use it as the best measure that we have in non-invasive neuroscience of excitability or connectivity, effective connectivity that can be probed in a directional manner. Right? If I was looking at the N100, there's so many different sensory confounds, and it's hard to, probably not a direct cortical propagation. There's probably some system level downstream effects. And so I just like to use it as a measure of excitability, if that makes sense.
So would it correlate with like a motor threshold or with E-field model locally?
Yeah, that's a good question. We haven't done those studies, but I think we should, certainly with the E-field. The motor threshold, how the motor threshold in the motor cortex relates to the excitability in a different brain region. I think people have explored that. I probably should know that literature better. But certainly looking at the E-field propagation compared to the local and downstream P30 amplitude, I think would be an interesting study to look at. And I would guess that there's some relationship.
Okay. So
the E-field in downstream regions, right? If you
Yeah.
look in the parietal region or medial region, the E-field's probably not going to reach that. So there, the P30 is picking up propagation patterns,
Yeah.
whereas the E-field may get only more local approaches. But I would expect around the DLPFC that there's a relationship between E-field strength and P30 response in the DLPFC.
Okay. And so the part where you personalize for not just modulating the P30, but for like the person's illness, right? With their symptom profile, would that personalization in your mind come around where the target that you stimulate, or with the pattern of stimulation that you use to move the P30?
Yeah, I think it's going to be a combination of everything. So, if you look at a different disorder, an understanding of which brain structures and which brain networks are abnormal, and in which way are they abnormal, then you need to figure out how to boost that person's, let's say, P30 propagation pattern between region A and region B to evaluate that brain network. And you can do that quickly without doing any sort of RTMS, long-term RTMS, but do single trains varied in this multidimensional space to understand how you can push that P30 around in an acute manner. And then you can pick that stimulation parameter set and apply that in a repetitive treatment manner. So it's a combination of knowing what circuit is going to be abnormal given the disorder. Our approach isn't really evaluating the brain-wide connectivity patterns and what's abnormal for that individual. But knowing that disease, knowing that you need to increase the frontal parietal network connectivity, or some network, once you have that network, I think you could take an approach like ours to say what stimulation patterns are best, and then update that in real time. It may be best on one day, but not best on the next day. And so I think that's where the adaptive piece comes into play.
Okay. And since you're focusing on the P30, do you think you were maybe chasing something with more of a cognitive outcome? There's a meta-analysis on just traditional RTMS for treating depression, and generally it's not seen to move the cognitive scores very much, like a little bit on trails A and B, but generally doesn't move cognition, like the cognitive deficit, very much. Do you feel like your marker is kind of treating that system better, or have you seen in AMIT's data that there's a cognitive improvement?
Yeah, another good question. There's a couple hidden agendas of the P30 approach. The first is that I think this approach can be used beyond just depression. Even in healthy controls, we don't have a toolbox to know how to increase or decrease the connectivity of any given brain circuit. We're sort of at a loss, right? And so I think outside of clinical depression and symptoms and cognition, using this P30 as an outcome measure to understand how you boost and weaken a brain circuit, even in healthy controls, can be a nice tool set that we can add to neuroscience as a whole. The second piece is we're exploring other markers outside of the P30. We're exploring other potentials, other peaks in the TMS EEG signal, certainly the N45 and the P60, anything below 100 milliseconds. After 100 milliseconds, it gets murky, as everyone knows, with the sensory potentials, but there still may be valuable information there. So I give this pitch as the P30, but we're also not putting all of our eggs in that basket. We're also exploring resting EEG. There's still that little bit of me that hopes that resting EEG is a valuable and potentially more effective biomarker than single pulse TMS EEG. Most of what I've seen is it's less reproducible. It's even lower signal to noise, and so I'm not putting that many eggs in that basket. But we can measure the resting EEG response in most of our studies in combination with the TMS EEG. And the last piece that I didn't present here is we're adding in cognition. We'll be adding in single trial markers of behavior that will target specific brain structures and networks. And so the combination with electrophysiology and behavior on a single trial basis, you can imagine that you could close the loop around each one of those. And people have done that in intracranial work, like Aliquid and others. And so the hope is that not all the eggs are in that basket, and there's a hidden agenda as well in just general neuroscience outside of depression.
All right, there's some questions in the chat. Do you guys feel like asking on your own? Valerie, you're first.
Should I read them, Desmond, or is Valerie going to?
I can ask, sorry. No, I'm fascinated. I was just wondering, because it sounded like you're talking about P30 being a generalized marker, but I don't know, it sounds like a pretty new literature. So have they recorded P30 to TMS stimulation when the stimulation was applied at a different scalp target?
Yeah, we're not developing the P30. The P30 or P25, some positive potential in that time period has been shown in motor cortex and other regions as well. So we're just choosing this broad, potential between, let's say, 20 and 35 milliseconds, the earliest peak that we can see. And it seems like that positive potential is there in the DLPSC as well as motor cortex. There's a negativity right after that, usually as well. And so we're also looking at that negativity to see if that's valuable as well. But it's really just the earliest potentials that we can see locally and in downstream regions. So 20 to 40 milliseconds, maybe that sweet spot. Earlier than 20 milliseconds, we don't necessarily have the hardware to reliably say that there's no artifact here. And certainly in more lateral regions like the DLPFC where there's lots of muscle artifact, this gets a lot trickier. In more medial regions, people can start looking in the teens of milliseconds and see nice responses. So we'd like to get there, but we can't. And I just don't look that much past 100 milliseconds because I just don't have that much confidence either in the mechanistic understanding of what that is or the confounds with the sensory off-target effects. So it's really just a function of finding that sweet spot of pretty early potentials that are reliable, that can measure some level of the network in a causal manner.
Thank you.
Yeah, good question. Miklos?
Hi, Corey. Very interesting results. My question was about this. I was just wondering, do you expect that there will be lots of variability in this tuning curve, especially about the shape? So it's like, I guess there will be some, in the absolute values, there will be differences, but in terms of the shape, that you will have completely different sweet spots for different individuals in the parameter space.
Yeah, it's a great question. My hope is that within an individual, if you moved around location in the brain, that you would see variability, that there's something about the DLPFC that maybe has this signature in this multidimensional tuning space. My hope is there's some consistency across subjects, but also some differences too, because if there was consistency across subjects, we wouldn't need to even measure this in subsequent subjects, right? We know that that tuning curve exists for the DLPFC, so we can just apply it, which would be great too. That usually isn't the case. And so, but I don't have any better predictions. I think in a few months, once these experiments are completed, I'll get you a better answer on that one. Good question. Greg?
Hey, Corey. Great work. It's really fantastic. I think you also did an amazing job laying out the problem space, and you have such a great view and approach.
Thank you.
You answered a lot of questions. The real challenge here is that signal to noise. You want anything earlier, and there's huge debates on whether you can get anything neural that's not just artifact from the TMS. 30 milliseconds is right there. Sounds like you're all in on the P30. Are you also collecting Licky, Sicky? Because ultimately they all have these established relations with plasticity, aberrant plasticity. Are you going for many, or are you just P30?
Yeah, it's a good question. Some of it is a function of our experiments and limits the ability to use these multiple different modalities in data collection. So in an ideal world, we'd like single pulse TMS, resting EEG, Licky, Sicky, all those sound great, even different locations of the brain, right? Contralateral and other control regions. Most of what we're doing is a crossover study comparing either different stimulation parameters in an event-related manner, so that we either can collect more trials or we can collect more modalities with fewer trials. And it's that trade-off. Some of our experiments are 5 hours long, looking at a couple different parameters. As you know, to get the signal to noise up, you have to get, we're doing 40 trials or 60 trials, and that's really the bare minimum. So in an ideal world, yes, we have some data where we have done Licky before and after RTMS. We haven't moved forward with that as much. I think the approach that we're taking is having single pulse TMS and then adding in a single trial measure of behavior, which is sufficiently different than the EFIS. I'd like to. I'm not dismissing those, and I think they would be very valuable. But when you think about the biggest bang for your buck, the single pulse TMS gets you the P30, the N45, and the P60, all with one pulse. Whereas the Licky and Sicky is more involved, and I don't have as much experience with it. But in subsequent experiments, it'd be really interesting to explore because, like you're saying, it does probe brain plasticity, and that's really what we're trying to do.
What is the behavior you're going after?
So we've been trying to figure that out, and I'm not a behavioral specialist. After hearing Alik's talk, we are thinking about like a multi-source interference type task. It can be single trial, it can activate the DLPFC, as well as the salience network and the frontoparietal network. We've explored other different types of tasks, but things like the NBAC require multiple different trials, multiple different stimuli in order to get a response. And if we really want a single trial, 1 second after the stim captures some aspect of behavior, that interference task may be a good one, and there's lots of literature on it. We're not reinventing the wheel or developing new methods around it. But I'm all ears. I'd love to hear what you think would be best to add in here.
We'll talk offline. I'd be happy. We're hiring at UCSD. For everyone on the call, if you're graduating any of your people at any level, we'll hire them. But fantastic work.
Thank you.
I'm ready, Greg.
Not you, this.
Helmet?
I don't want to keep. Yeah, Helmet.
Hey, just real quick, that was a great talk, Corey. I just wanted to follow up on the question about the E fields and the P30 that Desmond asked. How much do you think the P30 would correlate with things like skull cortex distances, things like the E field do? And then you mentioned that you see this as a more general marker, and I was just wondering if you see this as kind of a more intricate marker of dose for TMS.
Questions. So E field and P30. Yeah, I would guess if a brain is further away from the TMS coil, that there's going to be some change in the P30 that may correlate with the E field. I mean, the E field is capturing how much information is getting to that brain region. There's also local computation that needs to spit out the P30. So it may not be a perfect correlation, but I would expect some relationship. In terms of the P30 intricate marker of dose, compared to the 120% of motor threshold. Yeah, and people have been thinking about this. I know the Italian group and Finnish group, Marcello and Risto, they use TMS evoked potentials, 20 to 40 milliseconds, and they dose based on the amplitude of that evoked potential, and they feel very strongly that is how we should dose, and that captures the amount of information going in and that local computation that the E field does not necessarily capture. I think there's a lot of work that needs to be done before really adopting this approach, mostly around the artifacts that may exist around the muscle artifact and around the P30. So I'd like to get to that point. If we know more about this P30 or this time period, it would be nice to determine dose based on the evoked potentials.
Oh, well, I want to be respectful of your time. Really appreciate it, Corey. We had a much livelier discussion than we've had in a while, so I think everybody was excited to get back into the flow of these talks, and I hope many of you will come back and join us for more. Enjoy the last remnant of your time at home with the little guy, and definitely stay in touch.
Thank you. Likewise, and thanks everyone for listening. Really appreciate the questions.
Yeah. See you all next week.